In this slide I am going to explain the complete checkpoints of cell cycle during cell division. We start with G1 phase. This G1 phase checkpoint is activated when DNA damage is present in G1 phase and arrests cell cycle in G1 phase. Therefore, the cell cannot progress into S phase. In response to DNA damage, this protein gets activated, that is ATM or ATR protein, and activated ATM ATR protein activates P53 protein. This P53 protein synthesizes a CDK inhibitor that is called P21. This CDK inhibitor belongs to CYP or KIP family, and this CDK inhibitor inactivates this cyclin D CDK4/6 complex. Since cyclin D CDK4/6 is inhibited by P21, this cyclin D CDK4/6 cannot synthesize cyclin E protein. This cyclin D and CDK4/6 they synthesized in response to growth factors, and these proteins now is inhibited by CDK inhibitor, which is produced in response to DNA damage checkpoint. If there is no DNA damage, this growth factors synthesized cyclin D CDK4/6 complex phosphorylates a protein that is called RB protein, and this RB protein usually binds with another protein that is called E2F. Binding of RB to E2F inhibits binding of E2F to chromosomes. Binding of E2F to DNA causes transcription, and this transcription produces a cyclin E mRNA, and this cyclin E mRNA is required for the activity of CDK2. This cyclin E is required for activity of CDK2, and this CDK2 cyclin E complex enables cell to enter into S phase. So for this RB phosphorylation is important. So phosphorylation of RB enables binding of ETF to chromosomes, and binding of ETF to chromosomes synthesizes cyclin E, and this cyclin E is required for CDK2 activity, and cyclin E CDK2 complex can promote cell from G1 to S phase, and this cyclin E CDK2 complex is inhibited by in response to DNA damage at S phase entry. So in response to DNA damage, we know that these two proteins they gets activated, ATM ATR CHK1 and CHK2, and ATM ATR protein they synthesize P53, and this P53 in turn synthesizes P21, and this P21 inhibits cyclin E and CDK complex, and inhibition of cyclin A CDK arrests cell cycle at entry of S phase. Also this CHK1 and CHK2 inhibits this cyclin CDK complex by inhibiting CDC25. This CDC25 is a phosphatase. This phosphatase is required for removing inhibitory phosphates at tyrosine 15 and tryptophan 14 of CDK protein. Next checkpoint is in S phase. This S phase checkpoint gets activated in response to DNA damage. In response to DNA damage, again the same ATM ATR CHK1 and 2 they gets activated, and activated CHK1-2 inhibits CDC25A, which is a phosphatase enzyme. Since CDC25A is inhibited, it cannot remove the inhibitory phosphorylation of this CDK2 protein. In response to DNA damage, ATM ATR also produced in S phase, and this ATM ATR activates P53, and P53 activates P21 synthesis, and P21 inhibits cyclin A. As this cyclin A and CDK2 complex is inhibited by DNA damage, the cell is arrested in S phase, and it cannot progress into G2 phase from S phase. The next checkpoint is in M phase entry, or between G2 to M phase, and this is also activated in response to unreplicated DNA and DNA damage. In response to DNA damage, P53 gets activated, and activated P53 synthesizes P21. This P21 inhibits cyclin A, B, CDK1 complex. Also, the internal signal, that is unreplicated DNA, activates ATR protein. This activated ATR activates CHK1, and CHK1 in turn inhibits CDC25 enzyme, and inhibition of CDC25C by CHK1 inhibits cyclin A, B, CDK complex. This CDC25A is required for removing inhibitory phosphates from CDK1 complex. Since this cyclin A, B, CDK1 complex is inhibited by DNA damage checkpoint and unreplicated DNA checkpoint in M phase, this cell cannot progress into M phase from G2 phase. Cell is arrested at M phase entry. Next, two other checkpoints are there. One is in anaphase of mitosis, another one is telophase of mitosis. Two checkpoints are present here. One is chromosome segregation checkpoint, another one is spindle assembly checkpoint. In response to abnormal spindle fold formation, this MAD2 protein gets activated, and activated MAD2 protein inhibits anaphase promoting complex. This anaphase promoting complex, about this anaphase promoting complex I have explained in this video, and this anaphase promoting complex is very important for ubiquitination of securin protein and cyclin proteins of M phase. This MAD protein inhibits anaphase promoting complex by inhibiting this CDC20 protein, and this CDC20 protein is required for activity of APPC complex. Because the anaphase promoting complex is not active, this anaphase promoting complex cannot perform ubiquitination of securin. This securin protein proteolysis is required for activation of separase, and active separase only can separate chromatids. When chromatids are separated, then only the cell can progress into anaphase from metaphase. Since the separase enzyme is inactive, because of inactive CDC20 APPC complex, the cell cannot enter into anaphase from metaphase. Next checkpoint is in telophase. For progression of cell from telophase to cytokinesis, degradation of cyclin proteins is very important in telophase. This is the signal for cell to enter into cytokinesis from the telophase. Since this APPC complex is inhibited by misalignment of chromosomes and all, this APPC complex cannot perform ubiquitination. Therefore, the cell is arrested in telophase. Activation of anaphase promoting complex in anaphase is depends on CDC20, and this CDC20 is required for activation of anaphase promoting complex in anaphase. In telophase, the same anaphase promoting complex is activated by CDC14, and this CDC14 is required for anaphase promoting complex. If this CDC14 is inhibited, then this anaphase promoting complex cannot become active. Therefore, this anaphase promoting complex cannot perform ubiquitination of cyclins in telophase, and this CDC14 is inhibited when chromosomes are not segregated properly between two polar ends.
Hi viewers, this is GLN Prasad, Head of the Department of Zoology, Government Arts College, Anantapur. These video lessons are prepared by Dr. G. Rajasekhar, who happens to be the youngest faculty of Zoology Department. He has received his post-graduation degree from Hyderabad Central University in Medicinal Biotechnology. He has given plethora of information in these video lessons. These are like processed food and packaged food, which cater to the needs of each and every aspirant. I sincerely feel that the students will receive the right information that is useful for all the entrance examinations. I sincerely recommend these videos for all job aspirants who would like to get through all competitive examinations and other research examinations like CSIR, NET, APSET, NEET and MSET. I hope that these are the simplified versions of the most complicated topics. One can easily understand these lessons and also convert the same into multiple choice questions. They also increase your retention capacity, and you can easily grasp the content so that you can easily recollect while attempting the questions related to these topics in various examinations. One request from my side is, I sincerely request and I sincerely recommend these videos to be shared to all social platforms so that the students in other parts of the country and other parts of the state will have access to this valuable information. Wishing you all the best. Thank you very much.